Hi, I am Kairavee Thakkar from Dr. Nathan Salomonis' lab at Cincinnati Children's Hospital. I will be talking about our lab's computational tool, Udon, which involves patient subtype and phenotype discovery using single-cell genomics. Single-cell RNA sequencing has picked up pace in the last decade with its many technological advances. Meaning, originally people would sequence about hundreds of cells, and now there are methods that make sequencing thousands of cells quite efficient. And because of this, many people began analyzing from more than one sample and carried out analysis to show the differences between healthy and diseased samples. But then as the number of samples increase, it is challenging to identify the common and the patient-specific gene expression responses, especially in heterogeneous diseases such as COVID-19 infection. So there is a need in the field to identify novel patient subtypes that may not be evident from clinical features and associating these novel subtypes with the clinical parameters. So we developed a computational approach called Unsupervised Discovery of Novel Disease Programs, or in short, UDON. What we do here is that we start with the single-cell RNA sequencing data from each sample from a cohort, and based on similar gene expression patterns, we first identify cell type populations existing in the samples of the cohort. And this step is typically done outside of the program. But once we have this information, we will consider the cells from the healthy or the control samples only, and we compute the average gene expression from each cell type, which we refer to as pseudobulk. But for the cells of the disease samples, we follow a different approach of calculating the pseudobulk. Here we calculate the average gene expression of a cell type from each patient or disease sample. This is because we want to maintain the patient-specific expression from each cell type. We then compute the fold change or the difference in the pseudobulk values of the disease samples versus the healthy sample. So for example, we compute the difference between the monocyte pseudobulk value from patient 1 against the baseline monocyte pseudobulk value from healthy samples as a measure of how much change do we see in the expression profile of that patient's monocytes against the healthy monocytes. Now these fold changes between disease and healthy pseudobulks can be clustered based on their gene expression profiles using an unsupervised algorithm, and we get Udon clusters made of disease or patient pseudobulks. Each of these clusters are defined by their marker genes, which can be associated to a gene pathway using some gene pathway databases. In order to determine whether a clinical phenotype is statistically associated or positively enriched in a given Udon cluster, we developed a downstream approach called Satay Udon, which stands for Statistical Association Test for Clinical Phenotypes. In this approach, we test the null hypothesis that there is no association between a sample's clinical condition and the sample's pseudobulk profile in a given gene response program in a cell type specific manner. We applied Udon to a single-cell RNA sequence cohort of systemic juvenile idiopathic arthritis, or SJI patients, and SJI is known to be a clinically heterogeneous disease, and SJI is a chronic autoinflammatory pediatric disease whose causes are largely unknown to us right now. The single-cell data sets were collected by one of our collaborators, Dr. Grant Schuller's lab, and they collected the peripheral blood mononuclear cells, or PBMCs, from 26 kids, out of which 5 of them were controls. The 21 patients were classified into 4 clinical SJI categories: inactive SJI, active SJI, SJI with lung disease, and SJI with macrophage activation syndrome, or MAS, based on a set of clinical criteria. And these collected PBMCs gave us approximately 234,000 cells, which we are visualizing on a UMAP plot. And based on similarity of gene expression, we derived and labeled 30 clusters based on their marker genes and a tool called Azimuth that uses a PBMC reference atlas. When we applied Udon on this dataset, it determined 12 clusters. So here in the heat map, the columns are the patient or disease cell type pseudobulk profiles that are being clustered, and for each cluster there is a set of enriched marker genes, which are the rows here. Based on these marker genes, we identified the associated gene pathways, which included pathways that were enriched for interferon signaling, T-cell cytotoxicity, and complement activation. Previous studies suggested that interferon signaling is a driver for SJIA complicated by lung disease and MAS. One of the most distinct subtypes was Udon cluster 12, which was most enriched in interferon signaling pathway in active SJIA and SJIA with MAS. And this was typically seen in different myeloid and lymphoid populations. There was no Udon cluster that was composed entirely of pseudobulks of a single patient or of a single disease group, and cell types did not exclusively group together in the same Udon cluster. This indicates that the clusters are driven rather by gene programs expressed across cell types. Importantly, half of the Udon subtypes could not be identified using conventional supervised methods using differential expression analysis and comparative gene set enrichment. We also saw a novel gene pathway in Udon cluster 6 that was associated with complement activation. A subset of monocytic populations were associated with complement activation in SJI with lung disease and active SJI patients. And this was particularly interesting because previous studies showed elevated levels of complement components in SJI patients, but there was no study that had investigated the involvement of these complements in SJI with lung disease and the potential cell types that are associated with it. We were able to experimentally verify that complement activation is induced collectively in SJI with lung disease using ELISA on serum from 67 patients and controls. Here we are showing that C9 protein is significantly elevated in SJI with lung disease and MAS as compared to controls. These results were particularly interesting to us, and it motivated to ask us that we can visually see that the complement activation in monocyte is associated with lung disease, but does this hold true statistically? We applied Satay Udon and found confident associations between Udon cluster pathways and disease subtypes. We find that lung disease is strongly associated in the complement activation cluster for monocytic populations. Importantly, nearly all of these observations were unique to Udon clusters as opposed to clinically defined SJI subtypes. And these clinical associations can further be explained by cell type specific gene expression responses, such as the monocytic-driven interferon response and a novel platelet-mediated SJI inflammation through S100 proteins. To conclude, Udon and Satay Udon are powerful approaches to uncover novel shared disease programs from single-cell data of heterogeneous diseases, and we will also apply it to other patient cohorts of other diseases such as COVID-19 infection and autism syndrome. Thank you.